W22_Ms_4 extended 1. 1a Fig. 1.1 is a side view of a human skull indicating the 4 types of teeth in the jaws. I. State the function of human teeth. Mechanical, physical, digestion. E. State the name of the visible outer layer of the teeth. Enamel. E. Explain the process of tooth decay in humans. Any 3 from. Sugar, food, plaque, left on teeth. Respiration of sugar by bacteria. Produce lactic acid. Dissolves, enamel, dentine. E.g. dentine is exposed. Dentine is softer, dissolves more rapidly than enamel. Reference. 2. Pulp, nerve endings, being exposed or decay. Reaches nerve endings leading to pain. B. Mammals can be classified according to the position and shape of their teeth. Fig. 1.2 shows the skulls of 7 mammals. I. Use the key to identify each species shown in Fig. 1.2. Write the letter of each species, A to G, in the correct box in the key. E. Killer whales, Orcinus orca, are mammals. State 2 internal features you would expect to find in a killer whale that you would not find in a fish. Any 2 from. Double circulation, 4-chambered heart, lungs, any named part of lungs. Diaphragm, red blood cells without nuclei. E. State the name of the group of animals that includes mammals and fish. Vertebrates. 2 digestive enzymes catalyze the breakdown of large insoluble molecules. A. I. Explain why it is important that large insoluble molecules are broken down by chemical digestion. Any 2 from. Become soluble, for absorption, small enough, for diffusion, active transport. E. State the name of the substance that is the solvent for most molecules that have been digested by enzymes. Water. B. The activity of 2 protease enzymes, A and B, was measured at different pHs. Both enzymes are found in the human alimentary canal. The results are shown in Fig. 2.1. Describe and explain the roles of the 2 protease enzymes, A and B, in the alimentary canal. Use the information in Fig. 2.1 to support your answer. Any 6 from. Protein right pointing arrow amino acids. Reference to specificity. Both enzymes are active between pH 3 and pH 5, A at pH 4. A is pepsin, optimum pH at 2. Enzyme shows no activity, is denatured, from pH 5. Functions in stomach, where HCl is present in acid conditions. B is trypsin, optimum pH at 10. Enzyme shows no activity, is denatured, from pH 3. Functions in small intestine, secreted from pancreas. Bile neutralizes stomach acid in alkaline conditions. Breaks down proteins to polypeptides, peptides. Pancreatic juice neutralizes stomach acid. C. Maltase is a digestive enzyme that acts in the small intestine. State the exact location of maltase in the small intestine. Membrane of epithelium. 0610_W19_Ms_43. 3 carbohydrates are an important component of a balanced diet. The flow chart in Fig. 3.1 shows some of the processes that happen to carbohydrates in food that is eaten. 8. Describe what happens to carbohydrates in the human body between ingestion and assimilation in the liver. Use the information in Fig. 3.1 as a guide. Ingestion, digestion, described, occurs in mouth. Chemical digestion, absorption, described, occurs in small intestine, duodenum, ileum, ingestion of large biological molecules. Mechanical, physical, digestion, breaking, carbohydrate, food, into small pieces. Reference to chew, grind, bite, by teeth, tongue, swallow, moves through esophagus, churned in stomach, chemical digestion. Breakdown insoluble molecules into smaller soluble molecules. Salivary glands, pancreas, secrete amylase. Amylase breaks down starch, 2, maltose, glucose, sugar. Maltase is on epithelium of the small intestine, duodenum, ileum. Maltase breaks down maltose to glucose. Bile neutralizes stomach acid. Semicolon. Reference to neutral, 7/8 of a picohenry for amylase maltase, absorption into the blood via the alimentary canal. By diffusion, active transport, into villi, microvilli, capillaries. Microvilli, villi, folds, increase the surface area for absorption, increased blood glucose concentration. Insulin, secreted, produced, from pancreas. Insulin reduces blood glucose concentration. Reference to negative feedback, homeostasis described. 0610_S22_Ms_43. 4b, Fig. 4.2 is a diagram of a villus. The arrow indicates the direction of blood flow. Describe the structure of a villus and its role in the alimentary canal. Use the letters in Fig. 4.2 to support your answer. Any 6 from. P is a lacteal. Lacteals absorb fats, fatty acids, glycerol, named fat-soluble vitamin. T, lymphatic vessel, returns lymph fats to blood to circulatory system. Q, epithelial cell, has microvilli. Villi increase the surface area for absorption. Q is one cell thick for short diffusion distance, fast diffusion. Q is site of breakdown of maltose to glucose. Glucose uptake by active transport. R, capillary, for absorption of amino acids, glucose, salts, water, products of digestion, soluble nutrients. R has thin walls. R is one cell thick. Blood flowing to maintain concentration gradient. Transports absorbed nutrients to S, venule, vein. 0610_S22_Ms_41. 3. The pancreas is an organ that has roles in the digestive and hormonal systems of humans. Fig. 3.1 shows part of the alimentary canal and some of the associated organs. 8. State the names of organs J, K and L. J. Liver. K. Gallbladder. L. Duodenum, small intestine. B. The pancreas secretes hormones into the blood and enzymes into the pancreatic duct. The enzymes are released into the alimentary canal. Complete table 3.1 by stating the hormones and enzymes that are secreted by the pancreas. Hormones: insulin, glucagon, enzymes, amylase, carbohydrase, trypsin, protease, lipase. C. Chloride ions also move along the pancreatic duct. CFTR proteins in the cells lining the pancreatic duct move chloride ions out of the cells into the duct. Fig. 3.2 is a diagram of a cell from the lining of the pancreatic duct showing the location and activity of CFTR proteins. Explain how CFTR proteins move chloride ions across the membrane of the cell shown in Fig. 3.2. Any 3 from active transport against a concentration gradient from low concentration to high concentration. Protein changes shape to move chloride ions. Uses energy, e.g., reference, to carrier proteins. D. The movement of chloride ions into the pancreatic duct causes water to move from the cells into the duct to help the flow of liquid in the duct. Explain how water moves from the cell shown in Fig. 3.2 into the pancreatic duct. Any 3 from. Idea of presence of chloride ions in duct decreases water potential. Water moves by osmosis. Down water potential gradient from high water potential to low water potential. Movement of water through partially permeable membrane. E. If CFTR proteins do not move chloride ions, the liquid in the pancreatic duct becomes very sticky and the duct can become blocked. Blocked pancreatic ducts are one effect of cystic fibrosis, which is an inherited disease. Cystic fibrosis is caused by a mutation of the gene that codes for the CFTR protein. Fig. 3.3 shows the pedigree diagram of a family that has 2 people who have cystic fibrosis. The allele that causes cystic fibrosis is a recessive allele. Describe and explain the evidence shown in Fig. 3.3 that cystic fibrosis is caused by a recessive allele. Parents of 5 and 7 parents of people with cystic fibrosis 2 and 3 do not have cystic fibrosis. Parents 2 and 3 must be heterozygous carriers of the mutant allele. Person 7 is expecting a child with a man who is heterozygous for cystic fibrosis. Complete the genetic diagram to predict the probability of person 7 and the heterozygous man having a child with cystic fibrosis. Use the symbol A for the dominant allele and A for the recessive allele. 3e. Genotypes of offspring. Phenotypes of offspring. Probability of having a child with cystic fibrosis. 1 correct parental genotype A. 2 correct parental genotype A. 3 correct gametes from the parental genotypes. 4 correct offspring genotypes from their gametes parental genotypes. In any order. 5 correct offspring phenotypes and correct probability. MP1 and MP2 parents may be either way. Round but following rows must match. ECF from MP1 and MP2. ECF from MP3. ECF from MP4. Expected answer. Parent 7 man who is heterozygous. Parental genotypes AA. Gametes AA plus AA. Offspring. Genotypes AA. Offspring. Phenotypes without with. Cystic fibrosis. Cystic fibrosis. Probability 50%, 1 in 2 over 0.5. 0610_M20_Ms_42. 4.8 Describe the similarities and differences between marasmus and kwashiorkor. Similarities, max 3 from. Both caused by protein energy deficiency. Both types of malnutrition deficiency disease caused by lack of a balanced diet. Primarily affects children. Diarrhea, low body mass, weight loss. core growth, irritability, tiredness, wastage of muscles, dry skin, brittle nails, brittle hair, change of hair color, fatty liver, anemia. Differences. Kwashiorkor has water retention, edema, swelling of body parts, pot belly. People suffering marasmus also deficient in carbohydrates, fats, calories, energy. B. Researchers at a hospital recorded the total number of children admitted to the hospital between 1984 and 2002. A common reason for children being admitted to this hospital was severe diarrhea. Table 4.1 shows this information. I calculate the percentage decrease in the total number of children admitted to the hospital between 1998 and 2002. Give your answer to the nearest whole number. 18%, 3 megapixels. One selection of correct data, i.e. 1419 and 1161. MP2 correct calculation. MP3 correct rounding to whole number. E. Health workers in the communities near the hospital were trained in the prevention and treatment of diarrhea. This affected the total number of children being admitted to the hospital. Suggest the year in which the training took place. Give a reason for your answer. Any year between 1990 and 1994 or from 2000 onwards. The number of children admitted to hospital decreases after this year. E. The health workers provided advice to the community on ways of preventing the spread of the pathogens that cause diarrhea. Suggest the advice that was given to the community. Described hygienic food prep methods. Washing hands, hand sanitizers. Description of use of clean equipment. Use of clean boiled water, filtered water, pure water. Described examples of disposal of waste correctly. Idea of where defecation occurs. Defecation in a toilet. Monitor or identify infective individuals. Isolate infected individuals. 0610_W22_Ms_4 extended 2. 2 Fig. 2.1 is a diagram of the human alimentary canal and associated organs. A table 2.1 shows enzymes, the organs that secrete these enzymes, their substrates and products. Complete table 2.1. B. Explain the role of hydrochloric acid in the alimentary canal. Any 3 from reference to hydrochloric acid in the stomach. Kills bacteria, pathogens in food. Denatures enzymes in bacteria, harmful microorganisms in food. Provides acid, suitable, low, optimum, best, pH 4, pepsin. Protease, digestive enzymes, e.g. activation of pepsinogen. C. State a function of the region of the alimentary canal labeled 6 in Fig. 2.1. Reabsorbs water, ions, vitamins, e.g. fermentation of indigestible food matter by bacteria. Solidifying undigested waste. D. Describe how food eaten by humans is reduced to smaller pieces and explain how this makes chemical digestion more efficient. You may refer to Fig. 2.1 in your answer. Any 6 from mechanical, physical, digestion, breakdown. Decrease particle size, increase surface area of food for chemical digestion. For named enzyme activity. Any further detail of enzyme activity. Chewing, crushing, grinding, in the mouth, by teeth, amastication. Incisor canine, teeth for cutting food. Premolar teeth, crush food. Mixing of saliva with food, formation of bolus of food for swallowing. Churning action, formation of chyme, of stomach. 3. Reference to action of muscles in the stomach wall. Emulsification by bile, occurs in small intestine, duodenum, ileum. 5. Described as bile salts breaks fat globules into smaller pieces, large fat droplets into smaller droplets.